Okay, let's dive into a real medical mystery. This is a story about frustration, about years of searching for answers, and about a scientific breakthrough that's basically a genetic detective story for one of the most misunderstood infections out there. I mean, just think about that for a second. You feel sick. You've got these weird, persistent symptoms. Maybe it's fatigue, brain fog, joint pain, but every single time you see a doctor, the tests come back saying you're fine. It's a completely maddening situation, right? You start to wonder if it's all in your head. Well, for so many people who think they might have Lyme disease, this isn't just a hypothetical. It's their reality, sometimes for years, even decades. The standard tests just have these massive blind spots, and they leave a lot of patients stuck in this awful diagnostic limbo.
So, why does this happen? Why is this particular illness so hard to pin down? Well, this gets us to the heart of the Lyme disease dilemma, and why the usual clues that doctors look for, they just don't really work here. Here's the absolute key difference. The old tests, they're indirect. They look for your body's reaction to the bacteria, the antibodies you produce. But that reaction can take weeks to even show up, if it shows up at all. The new approach, though, is totally different. It's like a forensic investigation. It's not looking for signs of a break-in. It's looking for the culprit's DNA right at the scene of the crime. So yeah, the old methods are just full of holes. They often fail right when treatment would be most effective, at the very beginning. And even if you are infected, the bacteria that causes Lyme, this little corkscrew-shaped thing called Borrelia, can be hiding out in such low numbers in your blood that trying to find it is like looking for one specific grain of sand on an entire beach. And this is where the story takes a turn. Scientists knew they needed a totally new tool, something way more powerful, something a crime scene investigator would use to find that one single grain of sand, that tiny piece of bacterial DNA, and prove, without a doubt, that it was there. Enter PCR. This is the big breakthrough. The absolute easiest way to think about PCR is like a molecular photocopier. It can find one tiny, single fragment of the bacteria's genetic code in your blood and then make millions or even billions of identical copies. It takes something that was totally invisible and makes it completely impossible to miss. So here's how this whole genetic hunt works. First, you take a patient's blood sample and you isolate all the DNA inside it. Then you add these things called primers, which are like tiny, perfectly trained search dogs. They're designed to sniff out and latch onto only the DNA from the Borrelia bacteria. And once they find their target, the photocopying goes wild. The final step is just to see if any copies were made. And boom, this image right here, this is that aha moment of discovery. Each of those glowing white bands you see is a massive pile of photocopied bacterial DNA. So you can see the samples in lanes 1 and 2 are positive. That end lane, that's the negative control. No band means no bacteria. That bright band is the first piece of hard evidence. It's the smoking gun. But okay, just finding some DNA isn't quite enough. We need to know which type of Borrelia is it. Is it the usual suspect, or is it a more obscure relative? To figure that out, we have to move on to the next phase, actually reading the genetic fingerprint that we just found. What you're looking at here is the raw data from a DNA sequencer. Each one of those colored peaks is basically a letter in the genetic code. On the right, that's the unique fingerprint for Borrelia burgdorferi. That's the main cause of Lyme disease. But look at the one on the left. That's a different suspect, a related bacteria called Borrelia miyamotoi. You can literally see with your own eyes that their genetic fingerprints do not match. So how do we put a name to the face? Well, scientists take that genetic sequence and they upload it to GenBank, which is pretty much a global criminal database for microbes. And as you can see, the computer runs a match. The result? A 100% identity match. There is zero doubt, case closed. The suspect has been positively identified. So, this is all really cool lab stuff, but how does it actually play out in the real world? Well, this isn't just some neat trick. This technology was used to solve these long-standing medical mysteries for patients who had been suffering for a very, very long time. So the researchers in this study took this test and applied it to 14 patients. And these weren't just any patients. These were people with chronic, unexplained symptoms, and get this, they were all tested in February in the northeastern United States. We're talking the middle of a snowy winter. A brand new tick bite was pretty much impossible, which strongly suggests that their infections were old, not new. And right away, they got a huge surprise.
Out of those 14 patients, 4 of them tested positive, but not for the typical Lyme bacteria. Nope. They had its lesser-known cousin, Borrelia miyamotoi, an infection that the standard test would almost certainly have missed completely. And just look at this table. This is the real human story behind all the science. Patient number 9 had been dealing with symptoms for 20 years before this test finally gave them an answer. Patient 5, 15 years. Patient 14, 13 years. For these people, this test wasn't just data on a screen. It was the answer they had been desperately waiting for decades. But the biggest discovery of all, the one that really shows you the incredible power of this technique, was hidden inside the results for patient number 6. Okay, check out this genetic sequence. See how some of those colored peaks look messy? How there are 2 colors stacked on top of each other? That's happening because the machine is literally reading 2 different DNA fingerprints at the exact same time. This was the first time in the world that DNA sequencing proved a patient was co-infected with both the main Lyme bacteria and B. miyamotoi at the same time. I mean, what a groundbreaking find. So, what's the big picture here? What's the major takeaway? This is about so much more than just Lyme disease. This really represents a whole new era in how we can diagnose infectious diseases. You know, the researchers themselves said it best in their paper. They basically argued that tests like this, ones that are super sensitive and super reliable, they need to be put into practice in clinics and hospitals, especially in areas where these diseases are common, so we can finally give patients the fast and correct treatment they've needed all along. And that really leaves us with a pretty incredible thought. This technology gives us the power to read the DNA of an invisible enemy from a single drop of blood. It solved the mystery for these patients. The real question now is, what other chronic, unexplained illnesses could this kind of genetic detective work finally crack open?